factor B, factor D, factor P associating to our surfaces. Factor I, which is also on the surface of our cells, will bind any C3b that's deposited on the surface of our cells and cut it into I, C3b, which now has no known activity, and it just gets recycled as a serum protein by the liver. C3b binds to the surface of our cells. Well, factor H will compete with B. So this alternative pathway, this automatic happens all on itself, has multiple ways within our body that will help it to disassociate from our cells and keep things from progressing. We see that when it comes to the different steps in activation of these pathways, there are numerous proteins that are functioning within our body, whether you're talking the alternative, the lectin, or the classical pathway. The initiating proteases, serine proteases. Here, serine protease means it cuts at certain serines only. Factor D, MASP, and the C1s, remember that was a protease, are relatively homologous when you look at C1s and MASPs. They have some of the same structures, some of the same amino acid sequences. C3b, C4b bind to the cells. We see that also they're homologous in their activity. BB, C2a, homologous. All of them utilize C3b to begin opsonizing the surface of the pathogen to be destroyed. Initiating factors, C3b goes to activate C5, C3a. These begin inflammatory responses, also identical whether you're talking alternative, lectin, classical, because remember, after the C3, they are all the same pathway. It's the initiation between the three that will be different. So what we find is that C3b bound to the surface acts as a stable base once it's been converted by C3 convertase, not its own little autolytic conversion, but C3b bound to C3 convertase acts as a stable platform for the recruitment and the cleavage of C5. Now, here's the thing. To get C5 recruited to the surface of a pathogen, you have to have high numbers of the C3b there, which means you have to also have the high numbers of C3 convertase bound there. Once these are bound, and here they're showing from the alternative pathway with they stay bound and stable due to the different protein factors, you get minimal amounts of C5 recruited and cleaved. Turns out that those parts that are cleaved and then just released, they also have roles. They are not just floating away. They also have roles in initiation of an immune response. Depends on what is bound to them. Different fragments will be bound to and then taken up by different cells, which will make those different cells do different things. The different proteins, C3b, C3d, C3d, IC3b, that's when activated C3 gets cut up. C5a, C3a have specific receptors just for them. CD35, CD21, MAC1, also known as CD11b, CD18, or complement receptor 3, complement receptor 2, complement receptor 1, C5, C3a receptors. Once they bind to the receptor on one of these cells, they initiate different things. C3b, if it doesn't bind to the surface of a cell, but actually binds the complement receptor L large, will induce phagocytosis. If it is bound to the surface, its opsonizing surface of the bacterial pathogen, it will induce phagocytosis by a resident or a tissue-resident macrophage. So instead of being opsonized by antibodies, it can be opsonized by C3b. Aids in uptake. C5a binds its receptor, activates a G protein. Activates turns up the responsiveness of endothelial cells, mast cells, different phagocytes like the dendritic cells. Causes them to now go hunt, search for. So what we see is, however, whichever of those three pathways, mannose binding lectin, classical alternate, you end up with C3b bound on the surface. Well, it can trigger the rest of the cascade, and it can also trigger a reaction of a neighboring macrophage, because C3b will bind to C3b receptor, which will trigger the cytoskeleton to change and then wrap around and engulf whatever's bound to it via the receptor. The C5a promotes even more activity of the cell, of the macrophage, causes it to be more hyperreactive, makes it go out and hunt for more. The cleavage products are also recognized by different complement receptors. C3b either will deposit on the surface, as they're showing here. It's being left by C3 convertase. Brings in different factors that will help keep it there or will release it. Here it's factor I is releasing C3. B, it's cutting it, releasing it off to do something else. Well, the remaining parts of the C3, if they're still bound there, can cause a neighboring phagocyte, neighboring macrophage to cleave off parts of it, clean up whatever could be there. When you're looking at the neighboring capillary beds, you get the fragments that are not directly involved in the cascade. C3a, C5a, C4a will actually interact with the endothelial cells lining the capillary bed. Here in the lumen is the blood flowing through. Here is the tissues surrounding the capillary. C3, C5a, C4a will cause these endothelial cells to change. Here you see they're swelling up. There are now gaps in between the cell. The gaps allow for fluid from the blood to leak out. That's why at sites of an infection, sites of an injury, it swells up, because more fluid is leaking out in that localized area. With the fluid comes more complement proteins, comes antibodies that will help to opsonize anything not you. With the fluid going out and more of these that are not directly interacting with the endothelial cells leaking in, they will act as trail of breadcrumbs, also known as chemoattractants, chemical attractants, which will tell the different white blood cells, neutrophils, macrophages, T cells, this is where you exit out. This is where the injury, this is where the infection is. So exit out and begin working. Begin clearing out anything not you. Once C5 is cleaved and has deposited near the C3b, C3 convertase, you'll see the rest of the cascade, C6, C7, C8, C9, will quickly also begin forming their giant complex, each having their own job. C5b bound. Here the artist did not drew in the C3b, the C3 convertase. It's just assumed it's nearby. C5b, C6, C7 will interact with the plasma membrane. This stable interaction recruits in 8. Stable C5b, 6, 7, 8. 8 is actually inserts into the membrane, whereas C5, 6, 7 just bind to the outer surface. This stable starts bringing in more and more 9s. More and more 9s insert in and around, and before you know it, you literally have a pipe, a pore. Here they're showing a transmission electron micrograph of the surface of a cell that has these C9 pores embedded in it. Here they're showing you a lateral view, so you can see it actually is a protein complex that goes all the way into and through the membrane. This now becomes an open channel for everything inside the cell to leak out.